For our fourth and final lab exercise of the semester, we're going to be utilizing some ideas from chemical kinetics to actually solve a murder mystery. And so this is called homicide in the first degree. Our learning objectives will include the following. We're going to look at Newton's law of cooling, which is a first-order process, and use it to measure the time elapsed since an object began a cooling process. We're then going to analyze some first-order rate data to determine both rate constants and half-lives so that we can use them to similarly make a measurement of a time duration. We're going to use that rate data then to estimate the onset of a decay process involving biological molecules, and this will enable us to solve a murder mystery. So let's talk a little bit about the background. In this, the scenario is that somebody has been killed in their office, and you're called in to determine who actually committed the murder based on videotaped evidence. Okay, but they didn't videotape the actual murder, so what you have to do is perform a chemical analysis to determine the time of death so that you can narrow down the list of suspects. So what happens when a human being dies? Well, a lot of things happen, but in essence, living cell processes discontinue. And what that means is that the chemistry that goes on in cells is gradually going to shut down. Now, once that chemistry shuts down, there is nothing to keep the body warm, so the body cools during that time. We know that this is a first-order process, but as we'll see, there are some limitations to this process. As the body cools and the cellular functions break down, different functions phase out. And in fact, the organs of the body tend to discontinue their operation at different times. So it's not like you die all at once, but you die in phases, you die in stages. And the question that we want to ask is, do these stages provide us with a mechanism for measuring how long it has been since a body died, if no one was there to observe it when it happened? We're going to start by looking at this from the perspective of Newton's law of cooling. And what that law of cooling basically says is that hot objects cool according to a first-order kinetic process. So here's a description of how the temperature changes with as a function of time. Temperature is capital T and time is little t in this equation. Now the T with the subscript E and V is the temperature of the environment. So it's the ambient temperature of the location of the object that is cooling. And T0 is the initial temperature that it has. So as we can see from this equation, as little t goes to infinity, that exponential on the far right is going to go to 0. As it goes to 0, that means that the temperature of the body is going to approach that of the environment. And that's exactly what we would expect. As things cool down, eventually they reach thermal equilibrium with their surrounding environment. Okay, this process actually looks a lot like a first-order rate equation that we have from chemistry. Remember that the concentration of a reactant is going to equal its initial value times e to the minus kt. So we have that element here. The one thing that we have also, though, is that it doesn't decay to 0. It decays to a particular temperature, the temperature of the environment. Now for T0, we know that living humans typically have a particular temperature. It's 37 degrees centigrade. You may know it better as 98.6 degrees Fahrenheit. That's equal to 310 Kelvin. So we know there's a reference point at which we can refer this cooling process. And so, in essence, one can use that exponential decay equation that's given above to estimate the temperature since the last time a body had temperature of T equals T0. All right, now all I've done in this next equation is I've simply taken the top equation and solved it for KT. So you can imagine if we subtracted T environment from both sides, we'd have T of T, temperature as a function of time, minus T environment, and then divide that by T0 minus T environment, and that would equal the exponential e to the minus kt. And then if we took the log of both sides, we would end up with the equation given here. And I'll remind you that the log of e to the minus kt is just minus kt. Now, we can use this to determine the length of time that has passed when we measure the body at any given temperature. However, to do that, we need to know what that rate constant K is, and we don't necessarily know that off the top. And in fact, it's going to vary from one person to another, that rate constant. The other thing that's going to be a problem is that there is an inherent delay before bodies begin to cool, and this reduces accuracy. So I've put in an inset here at the very top of the diagram that shows the temperature of the body after death and how it cools. And you can see that if we fit an exponential decay to the long part of the curve, it actually comes up and hits the temperature of the body, that is 37 degrees centigrade, at a time that is before the actual time of death. And so that is what we would read as the implied time of death. And you can see that the implied time of death is always later than the actual time of death. The reason for this is that as the body is shutting down, it doesn't immediately begin to cool. In fact, it maintains its temperature to some extent for a little bit of time, and then it begins to cool according to this Newton's law process. So we're going to use this fact to estimate the time of death, but we know we cannot get an accurate time of death simply by measuring Newton's law of cooling. If we want to estimate the time of death, the first thing we're going to do is we're going to measure our times in minutes relative to midnight. Okay, the reason for this is that we can't just plug in a time like 2:30 a.m., all right? We've got to use a time that's an integer so that we can actually graph this and get meaning out of the rate decay. So what you'll need to do is if you have a time like 2:30 a.m., you need to convert that to a number of minutes since midnight. So each hour is 60 minutes, so 2 times 60 plus 30 would give us 150 minutes. So 2:30 would be recorded in our data as 150 minutes. We're then going to plot our logarithm of this complicated function of temperature versus time. Now, T environment is something that you would be given in this laboratory. So that'd be for example, it would be the temperature of the room where the body was found. T0 would be the temperature of the average human body when it's alive. The time, of course, is going to be this time measured in minutes. All right, once we do that and we plot this versus time, we're going to fit this data to a best fit or a least squares line. Now there's some instructions for how to do this in the appendix to the lab, and we'll talk about this in just a minute and I'll give you a little bit more detail. But you'll get a line that looks something like this. And from this line you're going to find the slope and the intercept of the line. Okay, the intercept is the place where the line crosses the vertical axis, and the slope, of course, is the slope that it has as it's decaying. The slope is generally going to be negative because the temperature is declining with time as we go further and further from death. Now, the slope, as I've indicated here, is equal to negative the rate constant. And so the rate constant is something we're going to need to find. And once we have the rate constant, I hope it's clear to all of you that you can get the half-life simply by taking natural log of 2 and dividing by the rate constant. That'll give you the half-life. All right, our initial time, t0, we can actually find from this data as negative intercept divided by slope. Now, I've given some mathematical reasons for this in the lab handout. If you want to learn that, that's great. If you just want to use the formula, that's fine too. This is the formula for finding that t sub 0. And that t sub 0 is the estimated time of death. So that's going to tell you roughly when the body died. But as we mentioned, since the bodies don't immediately begin cooling, that's only an estimated time of death. To get a reading, we're going to imagine a first-order decay process that involves the biomolecules. Now, I will tell you first off that this is a completely fictional means for measuring a time of death. This is not the way people actually do it. It is based on a little bit of fact and a little bit of hypothesis and speculation. However, this is what generally happens. In the human brain, it receives amino acids via blood flow. So one of the amino acids is tryptophan. Okay, and you can see the carboxylic acid at the far right of this. You can see the amino group that is one carbon removed from the carboxylic acid. And then it's got the side chain group, which is that fused ring system that involves a cyclic amine rather. All right, so the brain receives these amino acids, and what it does is it converts them into neurotransmitters, and this is happening via cellular reactions that occur in your brain. One of those neurotransmitters is serotonin, and it's shown here. And I've picked serotonin as one of several neurotransmitters that's created, but I picked it because you can see a little bit its chemical resemblance to tryptophan, and so one might imagine that there's a chemical process that goes on in your brain that converts one into the other. All right, now when blood flow ceases, that means that your brain is no longer getting amino acids, so the supply runs out. But this chemistry is still going on. Just because you die doesn't mean that the chemical reactions that go on your cells immediately stop. And in fact, they do continue for some time. So what one can imagine is that the amino acid supply runs out, the neurotransmitters continue to be built, but that will happen only until their supply of amino acids present in the cells runs out itself. So our supposition is that these amino acids can be measured relative to a known standard. Okay, we don't know a priori what the normal concentration of these amino acids are in the cells, so the reason we use a standard is to make up for the fact that we may not know exactly what the standard or what the time zero concentration of the amino acids might be. As they're used up, this concentration ratio is going to decrease following a first order process. So what I'm showing in this graph is a hypothetical decay of an amino acid, which I've labeled A. That's the blue curve there. And if I took that concentration as a ratio relative to a standard, which is a very slowly decaying chemical compound, you can see that the ratio of those two actually decays using the same rate constant as A itself. That is to say, they both have half decayed at the same time, so the half-life is unchanged. So that means that if we measure the rate constant of the ratio, we'll still get the rate constant of the amino acid disappearing, and that's the key to this lab. So how would we go about analyzing this? Now, I've written over onto the right-hand side the definition of the quantity that we're actually going to be measuring the decay of, and it's the ratio of the amino acid concentration to the standard concentration. So you might be given a series of points. You need to convert the times into minutes since midnight, so they're all going to be integers. And then you'll be given values of this ratio, and they generally won't be integers. Now, the first thing you need to do is to convert those values of R into log of R, because that's the thing that you're actually going to be graphing. Now, instead of graphing this out, I mean, you could graph it out, but what you really need to do is you need to analyze the data. You need to analyze the points that are all in purple in this table and use them to find the best fit line. I recommend actually using Wolfram Alpha, although you can use the Excel program to do this as well. In Wolfram Alpha, what you would end up doing is typing out a phrase in Wolfram Alpha that says linear fit, and then you would give the coordinates of each of the points. Now again, you're using the purple boxes in that table, so the time would go first and the number in the far right column would go second. So you can see that each of these points has been entered in here for Wolfram Alpha. Now when you hit return, you'll get a feedback from Wolfram Alpha that looks something like this. The very first thing is just basically confirming what it is you entered. So it's a good way to check and make sure you got all your data points entered correctly. And it's basically telling you have asked it to give a linear function. The very next thing it gives is the least squares best fit. This is the thing that you were trying to get from this analysis. So I'll point out that this is basically y equals this expression, 0.693249 minus 0.049904x. So from this, we would determine that the slope is equal to negative 0.049904. And that's the thing that's equal to negative k. So be sure to have that in mind when you get this feedback. All right, the intercept is that first number, the 0.693249. And I'll note that these are going to be different for each of the analytes. You're going to be analyzing 4 different amino acids and their ratios to a standard. And so you're going to have 4 different sets of data that you need to analyze. Each team member should do 2 of these so that you're getting a little bit of repetition, but also you're able to check against each other and make sure that you're getting accurate answers. If you have answers that don't agree, then you probably should recheck those and rework them. All right, now once we have all of this, What we're after is what is the time at which the ratio of this amino acid concentration to the standard was equal to its expected ratio. That's a number that you're going to be given in the lab worksheet. That number I've called R sub 0 here. So if we use that, using this linear fit, it would basically be equal to the intercept plus slope times the initial time. Okay, so the initial time is the one that gives you back this value of R0 at the time, that corresponds to a regular, normal, living human being. You can then take this expression and solve it for t0, and that t0 that you get would be the expected time of death based on this data. And this should be a more accurate reading than the one you would get from Newton's law of cooling. So this is the one you're going to use to solve the mystery. So as we go through the analysis procedure, the first thing you're going to do is simply use Newton's law of cooling to estimate a time of death. You're then going to analyze a bunch of amino acid analytes, and you're going to determine for each of them a rate constant for first order decay, and you should probably also calculate a half-life, because that gives you an idea of what sort of time frame is appropriate for that particular amino acid analyte. We're then going to use benchmark values to estimate a time of death, and that is going to be what you'll use then to compare against the video data so you can identify a prime suspect. So you really will get to identify someone that you think committed the murder in this particular mystery. Okay, as you're doing all of this, of course, you'll be completing various entries in your lab report worksheet. You will then be asked to answer some post-analysis questions, and I do expect you to work together on these. There are some challenging questions here, so I want you to put some thought into it and know that what I'm really looking for is to get an idea of your thought process. All right. Finally, I hope all of you will take the Lab 4 review quiz within a day or so of finishing the lab so that we can complete our work on this and I can issue your grades. I do wish you good luck. I hope you have a lot of fun with this lab. I think students in the past have enjoyed this because they get a chance to solve a mystery.